Alright, last time we met, we were discussing the ability of neurons to change their membrane potential from -70 because of their property of having electrical excitability, or of having that electrical excitability property. And we've discussed the differences between graded and active potentials. Today we're going to look at that in greater detail. In order for this to occur, we have to have the flow of ions, which we discussed occurs through various channels, which includes those that are shown here on this page that we talked about last time. Okay. We also discussed where that number of -70 comes from and what contributes to that resting membrane potential. Today we're going to see what happens when we move away from resting membrane potential. And so we'll begin discussing local potentials. Now, a couple slides ago, we had graded, and then in parentheses was local potential. So if you can't remember that those are the same thing, it might not be a bad idea to write that up on the top of the page. Okay, graded and local are the same thing. These are small changes away from resting membrane potential that allow for short distance communication. And they're much simpler than the action potentials that we'll get to later today. So reading through a small change away from resting membrane potential in a specific region, which is generally a small region of the neuron's plasma membrane, is called a graded or local potential. Okay, here in a second we'll look at an action or a local potential, occurring in the dendrites of a neuron. Okay. If the response makes the membrane, more polarized, which polarized means negative, so becoming more polarized, we call it hyperpolarization, or hyperpolarizing. Hyper means high or higher negativity, like we show right here. We're at -70, and then we're going towards -80. You guys would all agree with me that is becoming more negative, right? Yes? Okay. The opposite of hyperpolarizing is depolarizing. When you de something, it means taking it away, removing it. We're becoming less negative. Okay? So this is a depolarizing graded potential. Okay. This is only going to occur in response to a stimulus that causes those various ion channels that we talked about last time to open up. Okay. The ones that we're going to focus on are mechanically gated and ligand-gated channels. Things that can cause those to open up would be obviously a mechanical stimulus, like pushing in on your skin, okay, causing mechanically gated channels. Chemicals. All sorts of different chemicals. Things that we taste, things that we smell will stimulate depolarization or hyperpolarization. And heat can actually be a stimulus, and light. Of those, we're going to mainly focus in on a chemical stimulus, which on the next page here we can see two examples opening up what we call ligand-gated channels. Okay. Up at the top here, letter A, we're showing a mechanical stimulus. I'm not going to worry about what that mechanical stimulus is, but it causes a mechanically gated channel to open up. And look inside the cell. Here at rest, it's at -70, and you can know that with all these negative symbols. But when stimulated to open, the ions flow in. Look what happens to our membrane. It becomes positive. So it's not only becoming less negative, but it's becoming more, or it's becoming positive. So we would call that a depolarizing graded potential. Okay. The same thing is essentially occurring here, except this is a ligand-gated channel, which requires a ligand or chemical to bind to it, like acetylcholine. So we're going to talk a lot about acetylcholine in this class. Binds to a receptor that opens up that channel, allows ions to flow. We go from negative to positive, also depolarizing. Okay. But down here at the bottom we see hyperpolarizing, which means we're going to allow chloride in, when a ligand known as glycine binds to a receptor allowing chloride in. What charge does chloride have? Negative. So if we have negative coming into something that's already negative, we're going to go this direction, right? We're going to become more negative. So that's why it's called a hyperpolarizing. Here are 3 steps of local or graded potential, and we're going to use a chemical stimulus to describe what happens. Okay. Here we have a bipolar neuron. We said bipolar neurons, remember, have the cell body between 2 extensions. One's the axon and one's a kind of branch that then branches to the dendrites. And we're going to zoom in at just a small segment of one of those dendrites, and we see that its plasma membrane has ligand-gated channels that bind a certain ligand. We don't care what that chemical is. We'll just call it a chemical stimulus that binds to that sodium ligand-gated channel, allowing sodium to rush in. If sodium rushes in, are we going to become more or less negative? Sodium has positive charge, it's coming in, so we're going to become less negative. So that's why we would call it a depolarizing graded potential. So sodium moves down its concentration gradient, neutralizes some of that internal negative charge of the cell, membrane becomes, or the membrane potential increases. We could say it becomes more positive, we could say it becomes more negative, or less negative, excuse me. Just kind of how you want to word it. Okay. So instead of at negative 70, maybe now we're at negative 60. Does that make sense? Yes, it'll get there, I promise. Now, when sodium rushes in, sometimes just a little bit of sodium will rush in. Sometimes more, sometimes a lot more. We can see that here with our 3 graphs as a result of 3 different stimuli. Here, a very weak stimulus only allows a small graded potential to occur. Very slight depolarization, and then return back to resting membrane potential. Here, if the stimulus is a little bit stronger, we depolarize a little bit more and then return back to resting membrane potential. And then a strong stimulus, stimulus 3, we become even more positive or less negative, but then we return back to resting membrane potential. So these graded potentials can vary in amplitude, means that their height will vary depending on the strength of the stimulus. Okay. Graded potentials will also perform what we call summation. Think of summation like sum. When you're doing math and you calculate a sum, what are you doing? Addition, right? Adding stuff together. That's what summation is. On the previous page, we showed what happens if you do a stimulus and then let the graded potential die off, what we call decremental conduction, where it returns back to resting membrane potential. If you zap it with another stimulus before returning back to resting membrane potential, if these 2 stimuli are the same strength, you would think that the second graded potential would top off at the same point. But what actually happens is pretty cool. The second stimulus results in an even greater movement from resting membrane potential. We become even closer to 0 or positivity. We're becoming even less negative in this case. That's what summation is. The process by which graded potentials add together. Now, if a graded potential occurs in dendrites or cell bodies of a neuron in response to a neurotransmitter, we call them postsynaptic potentials. We'll talk about a postsynaptic potential later today. That'll be kind of the last thing we get to talk about. We'll talk about receptor potentials and generator potentials, which happen in sensory receptors and sensory neurons, in our lecture on sensations. So we'll kind of come back to those later in the semester. Why this is important is because it's going to get us closer to a concept called threshold. And threshold is something that is absolutely essential for an action potential to occur. And this is where the real magic happens. Action potentials or nerve impulses are brief but large electrical depolarizations and repolarizations of a neuron's plasma membrane due to the opening of voltage-gated ion channels. Unlike a local or graded potential, an action potential can travel along the membrane away from its site of origin by a process we call propagation. Propagation and conduction you can kind of use interchangeably. So you'll see conduction later today as well. Okay. When I say depolarization, that means becoming less negative, or that might actually end up becoming positive, which we'll see today. So what do you think repolarization means? It's going back to negative. So what we're going to see today is this and then this. We can see that back here on this page. Okay. Going like this, becoming less negative, is the depolarization aspect. Going back down towards negative would be the repolarization. With action potentials, they're not going to be just little bumps up and down. They're going to be big up and downs. You'll see that here on this page. I already mentioned this concept called threshold. I like to think of threshold as opening up the floodgates. Which at that point, when you open up the floodgates, whoosh, everything rushes through, and in this case, we're letting ions in and out of the neuron. And that is going to occur at a very fast rate, and that's what causes an action potential to occur. A threshold in neurons is what we call negative 55, or what number is negative 55 millivolts. Okay, represented with this dotted line. Okay, if our local potential is really weak, what we would call a subthreshold stimulus, we haven't reached negative 55, have we? So what happens is it returns back to resting membrane potential really quickly. If we have what we call a threshold stimulus, which is much stronger than a subthreshold, look what happens. We hit negative 55, and then boom, we become positive, and then boom, we return back to resting membrane potential. It's a very fast depolarization and just as fast repolarization. Okay. If we have a suprathreshold stimulus, it's not the same as we saw here back when we could add or summate. It's not like it's going to go up and up and up and up and up. We're going to create an action potential, depolarize, repolarize, back to resting membrane potential, on and on and on again. The stronger the stimulus, the faster we will create action potentials. Okay, if it's a weaker suprathreshold stimulus, these will be spread out a little bit faster. Or a little bit more, I mean. I know this is sounding really funky. I promise it'll start to stick. We can describe an action potential with what we call the all or none principle. Okay. If a stimulus depolarizes a neuron to threshold, the neuron is going to fire at its maximum voltage. Okay. It doesn't matter if it's just barely hitting negative 55 because we're just at threshold, or if we are really strong with our stimulus, the end result is still the same. We top off at the same spot no matter what, about positive 30, 35, somewhere in that neighborhood. Okay. If we don't have enough strength to reach threshold, the neuron's not going to fire at all. So it's all or none. Does that make sense? The great thing about summation, though, if we have a whole bunch of subthreshold stimuli, remember they will summate, 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 and then finally we will create an action potential because we will finally hit threshold. Okay. So if you have several subthresholds, a singular threshold or a superthreshold, the whole end result is going to be the same. Action potential, depolarization of 30, repolarization back to negative 70. Let's talk about what actually happens during an action potential. 7 things are listed here. It might not be a bad idea. It's not titled at the top, but these are the steps of an action potential. I would advise that you write that down. I cannot stress this enough to you guys. This is absolutely important to know. I will not do this on the test, but a great way to know if you know it, see if you can take out a blank piece of paper and write out all those steps. I won't ask you to do that on a test. If you can do it, though, you know the stuff, and that's great, because I will ask several questions on the test. Now, these 7 steps coincide to what's written here on this page. Okay, looks like some of you guys are still writing, so I'll bounce back. This is a graph of the changes in voltage that occur during a singular action potential. Okay. We're going to use a chemical stimulus creating a local potential. That's our step 1. It doesn't have to be a chemical stimulus, but that's what we used a couple slides ago, so we'll stay with that. Chemical stimulus binds to a sodium ligand channel. Sodium rushes in. If enough sodium rushes in that we reach threshold, now we've opened the floodgates. Step 2 is where we open what we call voltage-gated sodium channels. And they allow sodium to rush in at a very fast rate. Look how quickly we become all the way up to positive 35. Okay. Now down here at the bottom it says time. This is in milliseconds. This is not minutes, hours, seconds. This is almost instantaneous. Sodium is going to rush in at a very fast clip. But then we shut those channels. So at point 4, the sodium channels become inactivated and closed, which then allows for what we call voltage-gated potassium channels to open up, which then is going to allow potassium to rush out, which is down its gradient. As potassium rushes out with its positive charge, what's going to happen to the inside of our plasma membrane? Inside of our cell is going to be returned back to negative, right? So look what happens. We're repolarizing. We reach negative 70. We're back at resting membrane potential. But we actually, and it's not accidental, it happens just because, we don't stop there at negative 7. We actually dip a little bit past it. And so we become hyperpolarized. Remember, hyper meant higher, polar meant negativity. So we become even more negative. It's not a huge dip, but it is a little bit of a dip. But then a few milliseconds later, we bounce back out to negative 70. So like I said, you better know those 7 steps. Won't make you write them all out. I might even ask you to explain what's happening in the various parts of this graph. This is not a graph that you're going to find in your textbook because this is what they have in the textbook. Which is not a bad graph, but I kind of like the other one better because it breaks down the steps and gives you numbers to go along with them. Here they're showing you with little regions that are color-coded. This kind of shaded purple region, lightly shaded purple region, is where the neuron is at rest. Here a stimulus causes some channels to open up. Local potential occurs. We reach threshold. Sodium channels are open at a high clip, voltage gated that is. Sodium rushes in. Somewhere around positive 30, 35, we top out. Those voltage gated sodium channels close. Potassium channels open. Potassium rushes out. Okay. We cross back to negative 70 and past that, illustrating our hyperpolarization phase. Until finally, a couple of milliseconds later, we reach resting membrane potential. What's amazing is this is going to happen on every segment all the way down our axon. All these segments of our axon are going to have voltage-gated sodium, voltage-gated potassium channels. Think about the hand on the hot stove that I've been talking about all semester long. For us to put our hand on a hot stove and remove it within less than a second, probably, we have to create an action potential that spreads up our arm. And then in our spinal cord, we create a new action potential in another neuron. And then we have another neuron, there's probably several neurons, that come out with an action potential running to our muscle, which then causes our muscle to contract, which is a whole host of other things that we'll talk about. So this stuff that happens, we haven't got to all the stuff, is amazingly fast. Now, here in this figure, you can see they've got this green and tan region identified as a, what we call, absolute refractory period, and then this region here, what we call the relative refractory period. We'll talk about what those things are here in a second. Here's another figure to show essentially the same thing, but focusing in specifically at the voltage-gated channels. At rest, both of them are closed. We're at negative 70. If we reach threshold at negative 55, the voltage-gated sodium channels will open, sodium will rush in, and we become very, very much so less negative, all the way to positive 30, 35. At that point, the voltage-gated sodium channels close. We can then repolarize by allowing potassium out by way of the voltage-gated potassium channels. We repolarize back towards negative 70, and then we can start the whole process over again, or we can just return back to rest. Okay. This whole process, like I said, is going to spread like a wave down our axons, like we can show right here with this figure. Okay, we're zooming in at just one small segment of axon. What we're looking at is the plasma membrane. Okay, this segment of our plasma membrane is in the depolarizing phase. Sodium is rushing in through the voltage-gated sodium channels. A little bit of that sodium will scoot over here, which means this segment will reach threshold, which then allows for sodium to rush in really quickly. And then some sodium will scoot over here, and then that will depolarize the threshold. And then an action potential occurs. And so on and so forth, all the way down. As depolarization spreads down, behind it, repolarization will spread down. Because what we need to do is get the axon back to resting membrane potential so that it can fire again. So in theory, an action potential can be spreading down an axon while another one is starting to spread down because it's happening so quickly. Okay, because if we're still stimulating that neuron, it's going to continuously create action potentials. But it will only do so as long as it returns itself back to resting membrane potential first. That's what the refractory period's referred to. How about that transition? Refractory periods, and there are two different types, absolute and relative. Refer to the time, and this is just a few milliseconds, during which you cannot stimulate an action potential to occur, or it's really, really hard to. Okay. I came up with this analogy just a little bit ago, and I think it works pretty well. Let's look at the figure to show what I'm talking about. Okay. This is just one segment, might be right here at the beginning of our axon, at the trigger zone. We're in an action potential right here, aren't we? Depolarization, repolarization phase. Is it possible to start that all over again? No, not until we reach resting membrane potential. Okay. If we're using my analogy of jumping, and you're in midair, can you jump again? If you're falling back down to earth, can you jump again? When can you jump again? Right when you hit the ground. But in order to do so, you got to catch yourself, right? It's very kind of difficult to jump without a little bit of a pause in there. Okay. During the time which you're in error, we can think of that like the absolute refractory period. I always tell students to remember during the absolute refractory period, you cannot create an action potential again. It's absolutely impossible. No stimulus of any strength will trigger an action potential during this time. Relative, it is possible, but only with the unusually strong stimulus. So it's like when we hit the ground, if you're really good, you can jump again. But for most people, you gotta catch yourself. Okay? Regain your balance and jump again. Okay. Why does it take a really strong stimulus? We'll think about where we're at this point right here. What was our floodgate? Threshold, right? We're farther away from threshold now than at any point we've ever been. So to get back here has to be a real strong stimulus. The great thing about absolute and relative refractory periods, they are short, because we're talking milliseconds here. Does that make sense? I know, it's going to sound real murky to you guys. It'll get there, I promise. I've already mentioned this next stuff, what we call propagation. I've been talking about it, even shown a figure to illustrate it. This ability for neurons to spread that action potential down its axon from its trigger zone all the way to its axon terminals. Remember, neurons, and I'm just going to draw a simple one up on the board, have this segment right where the cell body tapers to become the axon, and that's called the axon hillock. Right in there is what we call the trigger zone, which is where the action potential is first triggered, hence the name. Okay. When that action potential is triggered there, it's going to then spread down the length of the axon to these branches, which are the axon terminals, or telodendrons. Okay. Now, it does so either as continuous conduction or as saltatory conduction, and it depends on whether or not the axon has myelin. Continuous happens in unmyelinated axons. Saltatory happens in myelinated axons. Here we can see a figure to show off both of these. It's better drawing here than my terrible neuron over there, but that's okay. So we've got our neuron. This is a multipolar neuron. Right here in the axon hillock, I said, is the trigger zone where the action potential is first occurring. Okay, we're just going to show this depolarization aspect of things. But sodium's rushing in through voltage-gated sodium channels. That's at 1 millisecond. 5 milliseconds later, we've spread it down the length of the axon a little ways. By 10 milliseconds, we've spread a little bit farther. Think of this continuous conduction like lighting a match or lighting a fuse, where it's going to burn continuously from where you lit it all the way down. This is going to be a slower burn, if you want to think of it like a burn. Okay. Look at where we're at after 10 milliseconds here versus 10 milliseconds here, which is saltatory conduction. Saltatory conduction, think of it like skipping a rock on water. The action potential skips from node to node. Remember, nodes of Ranvier are those bare segments where the myelin is not present. And so that is something I talked about the other day. Besides insulating those axons, myelin helps speed up propagation. We're almost all the way to the end of our axon at 10 milliseconds when we're looking at our myelinated axon versus here we've only made it maybe a quarter of the way. So saltatory is much more rapid than continuous. Okay. And therefore, if the axon is myelinated, it will send signals faster than if it's unmyelinated. That is one of these three factors that affects speed of propagation. Okay, AP, short for action potential. Okay. As I mentioned, number one, we just talked about myelinated axons propagate action potentials more rapidly. 2, axon diameter. The wider the diameter of the axon, the faster it sends a signal. So I don't really know why. Never really researched it. But later, when we get into talking about sensations, we'll talk about A, B, and C fibers, which is just describing their size. A's are the largest. So there are certain receptors that are A fibers that send signals faster than B and C fibers. Okay. So we'll see that later. That'll be an introduction. And then temperature. The higher the temperature, the faster the speed of propagation, because it has everything to do with flow of ions. Same concept as when we talked about diffusion. How diffusion happened in faster and warmer temperatures, like putting color or food coloring in cold water versus hot water. Same thing occurs with propagation. Great thing is our body's warm, so that promotes fast propagation. So now we know how an action potential gets to the end of the axon. We get to talk about the fun stuff, and that's how that signal can get transferred from one neuron to another neuron or to another cell. That's what we call a synapse. The place where signals are transmitted from one neuron, what we call the presynaptic neuron, as we see here, we've got 3 different ones, stimulating, or it could be inhibiting one singular postsynaptic neuron. Okay. We'll just kind of focus in on this aspect. We don't really want to worry too much about those. Those kind of just get us confused. We know we talked about last time dendrites are the sites for receiving signals from other neurons, and that's because they receive signals coming from a presynaptic neuron, and then they are part of the postsynaptic neuron. Okay. This is going to illustrate what we call a chemical synapse. We'll talk just briefly about electrical synapses in the next slide. We'll get into those in greater detail later in the semester, and I'll tell you why here in a second. Okay. Now, this is an example where we've got a neuron stimulating or inhibiting another neuron. Okay. Notice next to postsynaptic neuron, I have /cell. We talked the other day about how the somatic nervous system is involved with voluntary skeletal muscle contraction. Autonomic is involved with involuntary control of glands and cardiac muscle, smooth muscle. Well, those various organs, skeletal muscle, heart muscle, whatever, they're innervated by neurons. Neurons that have the ability to send signals from them to their cells. So instead of a neuron being the postsynaptic neuron, it could be a muscle cell being the postsynaptic cell. And so we'll see that when we get to talking about muscle contractions. Instead of a neuron being here, it'll be muscle cells that are then being communicated by a neuron. First, let's talk about electrical synapses. And we'll just spend one slide talking about these for now, because we'll talk about them in greater detail later in the semester. These are really cool. It involves what we call gap junctions that allow action potentials to spread from one cell directly into a neighboring cell. A gap junction is just like a doorway between 2 cells. Okay. So like that door over there that leads to Mr. Muller's office, if we all walk through it, we can pretend that we're ions. Inside of this room, we've depolarized and created an action potential in this room, and then we walk into his room, we create an action potential in there. And then we can walk into the next classroom and create an action potential in there. Okay. This is awesome because it enables our heart, where you see this happening, to depolarize all of its cells in certain areas of the heart at the same time. Because when a muscle cell depolarizes, it's going to contract. So it enables our heart to contract in a very, very coordinated manner with all the cells working together at the same time, which maximizes its pumping ability. So we'll get to that later in the semester. For now, I want to just talk about chemical synapses. Chemical synapses involve the release of a chemical by the presynaptic neuron. Those chemicals are called neurotransmitters that will bind to receptors on the postsynaptic neuron, or if it's like a muscle cell, a postsynaptic cell. These neurotransmitters, as we'll see in a little bit, can either be excitatory or inhibitory. When they bind to receptors, ions will flow in or out of the postsynaptic neuron, which creates a new potential, what we call the postsynaptic potential. I mentioned that earlier. We talk about those later today. Here's that time. Now, I'm not going to go back to the image. I'll just draw it up here on the board. But between neurons, there's going to be a tiny little gap that exists. Okay, and those gaps are found between what we call the synaptic end bulbs, which are found at the end of each one of our axon terminals. And the next neuron. Notice how my dendrite here that's coming close to that synaptic end bulb. See how I left a little gap in there? That was on purpose. We can see that. I guess I will go back to it. There's a little gap in there between that axon terminal or that synaptic end bulb and that dendrite, between those bulbs and that cell body, or even between that bulb and that bulb. That gap is called the synaptic end cleft. Or synaptic cleft, not end cleft. Synaptic cleft. Okay. And we'll see a picture to illustrate what that is after this slide, which is going to explain all the steps involved in a chemical synapse transmission. You had to learn 7 steps earlier. Guess what? 7 more steps to learn. This is how we can transmit a signal from one neuron to another neuron, or to a cell, like a muscle cell or a sweat gland cell. These 7 steps, like I said earlier, you won't be required to know and write them out from a blank starting point. But if you can do that, it's going to really be beneficial to you. You'll know you know it real well. Those 7 steps are all illustrated here on this figure. What we've done in this figure is zoomed in at one of those little bulbs. At the very end of our axon are these synaptic end bulbs. Okay, and that's what this segment of our figure is. So here's our action potential spreading down to the bulb, which is step 1. As that action potential spreads to the bulb, it triggers the opening of what we call voltage-gated calcium channels, which enable calcium in to the bulb. Calcium triggers the movement of these bubbles, what we call synaptic vesicles. If you look inside these bubbles, there are these little red dots, which are the chemical messengers, what we call neurotransmitters. Those vesicles will move to the presynaptic membrane, which faces the cleft. And then they fuse with that membrane and spit the neurotransmitters out by exocytosis. Those neurotransmitters move into the cleft and across to what we call the postsynaptic membrane, which is, if you look at my figure over here, going to be just the tip of my dendrite. So those chemicals were released by the bulbs and will go across and bind to receptors on this case, or in this case, to my dendrites. And then they open up and allow ions in or out. In this case, sodium is going to come in. So we've had our action potential spread to that point. Okay. This guy, before that, was at rest, which his membrane potential was at -70. Guess what happens if sodium rushes in? We're not going to stay at -70 anymore, are we? What's going to happen if enough sodium comes in? We can hit threshold. Guess what happens when we hit threshold? We can create a new action potential. That occurs if we depolarize the new neuron. And that requires going away from negative 70 towards negative 55. And that requires excitatory neurotransmitters. Excitatory means that we're going to excite the neuron. Excite means that it might have an action potential. Okay. Excitatory neurotransmitters that are released cause both sodium and potassium channels to open. Sodium rushes in faster than potassium out, so we become slightly less negative. If enough sodium would come in, guess what? We hit threshold. Boom, action potential in our new neuron. Okay. We call this the EPSP, excitatory postsynaptic potential. There's also inhibitory neurotransmitters that make us go away from threshold. Okay. They cause things like potassium to rush out and or chloride to rush in. Well, if we lose potassium or we gain chloride, both of those are going to have the effect of making us become more negative. More negative means that we're farther from threshold and we're less likely to create an action potential in our new neuron. So that's why we call it inhibitory. Okay. This is an IPSP, inhibitory postsynaptic potential. We're far less likely to create and excite the new neuron or cell. A great example of how we use both excitatory and inhibitory happens as simple as when you go to the doctor and he taps on your knee. What happens when you tap on that patellar tendon? You kick your leg out, right? We have sets of signals that go to our quads that cause our quads to be excited and contract, because your quads are what kick your leg out. Okay. At the same time, we send inhibitory signals to our hamstrings, which are on the back of our thigh, because they do the opposite. They bring our leg back towards our butt and kick our butt. Okay, but we want to kick out, not back. So we're sending excitatory in one area, inhibitory to another. We'll talk about this later in this unit. That'll be our last lecture of this unit. Okay. Once again, going back to that hand on the hot stove. There's so much going on with at least 3 sets of neurons coming in, sensory, interneuron in the spinal cord, motor neuron going out, and then we haven't even gotten to muscle contractions, and that's complicated in its own right. All that happens within milliseconds. Maybe a second, hopefully not too long, so we don't hurt ourselves even worse. I hope by now you guys are appreciating how amazing the nervous system is. All right, we will